Welcome to the last talk of the 2015 NASA Ames Summer Series. We began the series with a talk entitled Brain Function Through the Eyes of the Beholder by Dr. Lee Stone. We went through a journey where we looked at earth science, materials needed for re-entry, science fiction on Mars. We also had talks as far from the Vatican Observatory. And so we went through different stages of what NASA does and what we see in the world. Today's talk, entitled Fractal-Based Mapping of Vascular Patterning for Human Space Exploration, will talk about visualization. And specifically, when we could visualize processes, we could understand them better. By understanding them, we could approach risk mitigation, and we could also look at new futures. The talk will be given by Dr. Patricia Parsons. Dr. Parsons received a bachelor's in science and engineering from the University of Michigan in 1983. She then continued her education and received a master's in science and chemical engineering from Case Western University in 1989, and a PhD in chemical engineering from Johns Hopkins University in 92. She joined NASA Glenn Research Center in 2001 as a biomedical engineer and joined Ames now in 2014. She is the lead innovator for NASA's VestGen Software. In this talk, she will describe VestGen, the process, and how it could be applied for space exploration. Please join me in welcoming Dr. Parsons.
Thank you, Dr. Cohen, for your kind introduction, for the opportunity to present our research today. Complex fractally branching vascular systems are essential for all higher life forms on Earth, including humans, other vertebrates, insects, and plants. Vascular systems in our body perform the distributed communication of essential metabolic and other factors between widely separated, specialized cells, tissues, organs, such as the heart, brain, and skin. In humans, for example, every cell in our body must reside within approximately 500 microns of a small, fragile capillary vessel to obtain necessary life support. Downstream of our single pumping hearts, approximately 40 branching generations of successively tapering vessels connect down to these tiny capillaries that are the only site performing the vascular transfer functions of exchanging the metabolic, immune, reproductive, and other factors that we utilize in our bodies. Our research discovery story started with my experimental observations at the microscope at the University of Washington, where I was a postdoc and then junior faculty. What I observed that was surprising to me was that every major regulator of vascular remodeling and growth induced a specific, unique fingerprint or signature vascular pattern. So these observations suggested the plausible long-term hypothesis we continue to pursue in our research is that vascular patterns can provide us with relatively accessible but integrative readouts of complex dominant molecular signaling, because as my space biology colleagues know much better than I do, these molecular signaling pathways are complex and really required the modern omics revolution to begin to dissect. So, when faced with the bewildering, amazing variety of vascular systems in the various species, tissues, length scales, one unifying principle that we really can hang on to is the usefulness of fractal branching as a natural space-filling mechanism in physiology. Our research story did start with the avian chorioallantoic membrane, or CAM. That was the research in which I developed the initial observations. Then, as funded by NIH and NASA, we extended these observations into the human retina for looking at disease progression and visual impairments in both terrestrial human populations and now astronauts. These human retinal stories are supported by other model systems in the mouse. Other systems we have applied the VestGen approach to include gastrointestinal inflammation, the venation patterning in the leaves of higher land plants, venation in the wings of the fruit fly, Drosophila melanogaster, which are a very important genetic model for drug discovery, and even to some extent in intravital blood flow. So the story that started at University of Washington and continued when I joined NASA in 2001 is now leading to our first studies in the retinas of the astronauts pre and post flight to the ISS, or International Space Station. NASA has recently identified, in the past several years, significant risks for visual impairments associated with increased intracranial pressure that are associated particularly with long-duration space flight. We're just starting these studies, and I think we'll have the results from them in about a year, ready for public disclosure. But it is really the studies that we did in the avian CAM and the human retina that provide the compelling preliminary evidence, as recognized by the peer reviewers in NASA, for initiating these astronaut studies. We have designed the VestGen software to automatically map and quantify vascular patterning from binary images, according not really to engineering principles, but according to physiological fractal-based branching rules that also involve considerations of anatomy, fluid mechanics, and cell biology. These branching rules that we follow include vessel bifurcations, vessel tapering, connectivity, and also account for the fluid mechanics of blood flow. The three fundamental types of vascular geometry listed here include trees, as illustrated by our map, VestGen map of the human retina, vascular networks, illustrated by the mouse retina, and then tree network composites, of which this really is an example. I've again listed here the various applications we have tested, published, and just to say that the ubiquity of the fractal branching phenomenon has been quite an interesting story that I wouldn't have predicted. This slide illustrates what I think you all know is the normal progression of basic to clinical research in biomedical studies. So initially, we might identify and propose feasibility candidates for drugs and other therapies by looking at molecules and cells in vitro studies. After identifying candidate strategies, these approaches will be tested and more complex in vivo organisms and ultimately applied in clinical studies in human beings. Now, the CAM model really represents a model of intermediate complexity between cells in a dish and something like the mouse or other rodents. because it's quite a simple system in that you simply have to crack the egg into a dish, in this case with quail eggs, a conventional 6-well culture dish. And then the little embryo and its chorioamnionic membrane, an analog of the human placenta, develop quite quickly over several days. You may well ask, why the quail egg? And the reason, as I've just described, is that it does grow very quickly. It is the chorioamnionic membrane, which is an outpunching of the gut, fills the petri dish and is easily dissected and fixed as a transparent specimen. Other conveniences of the model is that angiogenesis is very rapid and is contained within a 2D vascular tree that is microscopically accessible. The next two slides demonstrate for you your own observation, my initial observations looking at this system, and that the application of the major angiogenesis regulators, such as FGF2, VEGF, induced really the specific fingerprint or signature vascular patterns you see before you. My career research really does focus on angiogenesis, its stimulation and inhibition for health and disease applications. So what you see compared after 24 hours of treatment with FGF is that it was a simple, robust stimulator. It simply induces the growth of new, small blood vessels in a dose-dependent fashion. VEGF, surprisingly, was more complicated in its regulation. At low concentration, as FGF, it did stimulate physiological angiogenesis, or the growth of the new small vessels. However, at larger, higher concentrations, there was a phenotypic switch to a non-angiogenesis pattern in which the vessels were dilated, leaky, and associated with significant increase in the activity of endothelial nitric oxide synthase, or eNOS, a well-known vessel dilator. So when I looked at the major inhibitors of angiogenesis that include TGF-beta and angiostatin, and of course many other molecules, again, each of these regulators induced a distinct fingerprint pattern of response. TGF-beta, in a sense like FGF, targeted the growth of the new small vessels by inhibiting them, but maintaining the vascular tree in a normal state. Angiostatin, on the other hand, greatly disrupted normal vascular patterning, inducing what appeared to be a pretty pathological phenotype. So the prevailing hypothesis in angiogenesis research was established by Douglas Hanahan, Judah Folkman, Noah Bauk, and others, I would say, especially during the 1990s, and still guides research in the field. So this hypothesis states that angiogenesis, either normal and healthy in development or pathological in diseases such as cancer and diabetes, is turned on and off or off by the net accumulation of the many stimulators and inhibitors in our bodies. Our hypothesis, the VestGen hypothesis, builds on this angiogenesis hypothesis by proposing that fingerprint or signature vascular patterns really can provide useful, accessible, integrative readouts of these very complex interacting molecular signaling pathways, and of course even others such as hypoxia-inducible growth factor. To me, this is really the key slide of the entire talk. Because the observations and measurements that we made with this system continue to guide our research into other organisms and human disease. So the basic design principle of the VestGen software is to segment or dissect the branching tree into successive branching generations. so that we can measure where on the tree or how on the tree these complex changes are occurring. The approach that we take is that the first generation or parent vessel is identified, and then by fractal and other physiological principles, the remaining vessels are dissected into their respective generations. The reason I think this is the key slide is because, as documented by the complex quantification in a number of publications, including these 3 publications, we were able to supply scientific evidence in support of this hypothesis. So, to summarize by these sketches of the results, FGF did indeed act, as measured by our VestGen results, as a simple, robust stimulator, and that with increasing concentration of this cytokine, the increasing density of small new vessels were observed. VEGF as a vascular complexity factor did display these overlapping patterns of response. So at low concentration, the vessels, the new small vessels did increase as a physiological angiogenesis phenomenon. At higher concentrations, there was a phenotypic switch to a pathological, non-angiogenic phenotype in association with increased eNOS signaling, in which there is increased vessel diameter and leakage. TGF-beta as something of an inverse of FGF signaling with increasing concentration did strongly inhibit the growth of the new small vessels. So those results were mapped and quantified initially with the semi-automatic MATLAB prototype code that I wrote while at University of Washington. After joining NASA in 2001, I have to say it was my dream and my privilege to work with a brilliant computer science team consisting of a software architect and an image processing expert. So together we were able to fully automate the analysis into the types of mappings and quantification you see illustrated here. Using again, however, our CAM model as we matured the technology, with these cover illustrations, again, of the CAM branching generations within the trees. So to summarize this more mature phase of our research, we tested in collaboration with the Cole Eye Institute the effect of common clinical steroid use in clinics, not only for the retina, but for inflammation in other sites such as the knee. It's a very well-established anti-inflammatory and anti-angiogenesis drug, powerful in its inhibitory activities. So again, it was no surprise that we did measure and quantify the significant inhibition of the small vessels by this drug. However, because of the vascular patterning capabilities of our analysis, we did make, I think, what is a novel discovery, a useful discovery, in that the steroid also thinned the vessels, the diameters of the vessels, throughout the branching tree. This effect of the steroid, we thought, was related probably to its primary harmful side effect in the clinic, which is associated with increased risks for glaucoma. Glaucoma is a condition whereby the pressure increases to levels that it can be blinding. And so once we've disturbed the effectiveness of the vessels for regulating the fluids within the eye, it's possible to imagine how that could be associated with glaucoma. So finally, to conclude the CAM studies description that we performed, well, all of us in research know there are always the stories that get away and you just can't finish everything. So I just thought you might enjoy seeing this slide in which we tested an endogenous factor in our bodies that surprisingly resulted, compared to the normal control, it induced the development of very highly tortuous vessels, these little corkscrew vessels here. And of course, that also has implications for disease and health. Well, after seeing a number of slides of our CAM studies as our initial biological and engineering testbed model for developing the technology, I think you probably can all recognize in this slide the similarity of the branching trees in our own human retinas with the vascular patterns in the CAM. The CAM is often used in research as a morphological analog of the retina. And indeed, it was a former director of the United States National Eye Institute who suggested to us when I was in Washington that we use our CAM VestGen analysis for looking at human retinal disease. So these studies actually were done at the University of Washington with the Department of Ophthalmology. And here what we did was to take our CAM methodology, which was to assign the various portions of the retina according to the regions identified by the ophthalmologists, and then use the fractal analysis to measure and compare these regions of healthy human retinas with early stage disease and diabetic retinopathy. Diabetic retinopathy is the major blinding disease for working aged adults. So taking these approaches, we did see that indeed there was strong vessel dropout in the central region of the retina, the macula, which is very important for human and primate vision. And this slide actually was shown a few years later at the annual International Ophthalmology Research Meeting. And since then, a number of institutes in Australia and Singapore where vision research is very important because of the challenges to vision in the tropics, have established the fractal dimension as one of their ways of measuring vascular change with the progression of disease. Since the title of our talk is Fractal-Based Analysis of Vascular Remodeling, I thought I should spend a little time talking about fractal methods. So Fractal Mathematics was developed by Mandelbrot and others in the 1980s, and it's a non-Euclidean way of looking at many complex space-filling structures in nature, such as lightning, trees, coastline topography, vascular and neuronal branching. And I think some of you in the audience who focus more in the nervous system will pretty quickly see the similarity of branching in this lightning with neuronal branching in our bodies. A fundamental concept of fractal mathematics, probably the key one for branching structures, is the idea of self-similarity. So this is the phenomenon, the fractal phenomenon, whereby larger branching structures, such as you see up here, successively iterate that branching pattern at smaller and smaller length scales. So you will see down here another branching event that becomes smaller and smaller, but essentially repeats the branching structure. So just to say again that the pattern in the lightning is really rather different from vascular branching, which is water-based. But the patterns, when people ask us to use VestGen for neuronal branching, are really somewhat different, and I think have some kind of eerie connection with the branching in lightning because our neurons connect by electrical signaling. So looking at fractal algorithms in the late 90s when I started this research, I identified the box counting algorithm as the most reproducible and sensitive for detection of branching patterns in vascular structures. So this, you see here from this vascular structure that the box counting yields a fractal dimension of 1.41. So as defined by Mandelbrot and the other mathematicians creating this new mathematics, in a 2D binary black-white vascular image, the fractal dimension of a branching structure, such as the vasculature, must necessarily lie between the dimension of a line, one dimension, and two dimensions, such as in a square or a rectangle. So in this case, as we discussed in the previous slide, the fractal dimension will always be a fractional quantity defined by those limits. So now, after having examined the pattern, the concept of self-similar branching in the lightning photograph, I think you can now see how this concept of self-similar branching is repeated through about 8 generations of branching in the CAM specimen. Just to say that mathematical fractals, such as the Julia set, the Koch curve, those kinds of mathematical fractals can iterate themselves through infinite numbers of generations. But in nature, such as the vascular system, the self-similar patterns will typically repeat themselves up to generations of about, or iterations of about 8 to 10. So as I described in the CAM, looking at normal healthy development, which of course is a very important aspect of this kind of phenomenon, the fractal branching patterns quickly fill themselves in over about 5 days, 6 days of vascular development. And one result that continues to fascinate me from that early research is the fact that that fractal-based growth pattern becomes a linear result when analyzed by fractal mathematics. So you see here that growth pattern of the vessels is indeed linear during that developmental window. And this really is an exponential phenomenon analogous to talks we heard in our own space biology last week, in which the growth rates of microbes and cells also display a linear pattern of growth, because they are dependent, the rate of change is dependent on the current state of the system. So to summarize at this portion of the talk, just want to say that we have accumulated evidence in a number of systems about the fact that fingerprint or signature vascular patterns may offer biologists and physicians useful, accessible readouts of dominant molecular regulators and pathways. The system is fractal-based, but in order to really describe it, we need a number of other repertoires, a suite of repertoires, because at least in the current state of fractal analysis, which is an early stage in its development, really you can have the same fractal dimension for quite different vascular patterns. The dimension may describe the space-filling characteristics, but the patterns don't have to be the same. So it's really a non-unique descriptor. Therefore, to quantify changes in a site-specific manner within the tree, we have other parameters such as number density, length density, and branch points. So in collaboration with Dr. Peter Kaiser, vitreoretinal surgeon at the Cole Eye Institute, we did embark upon a new NIH study after I joined NASA. And so you see illustrated here the progression of this blinding disease from a normal, healthy retina to early stages and the late blinding stages when the vessels have overgrown and the neovascularization is blinding and very abnormal. So applying the methods and technology that we developed in the CAM to the human retina and the progression of this disease, you can see the maps of the arterial trees through mild, moderate, severe, and very late blinding stage, as well as in the venous Venus trees. So we were surprised to discover that there was actually a very strong regeneration of the small vessels as measured by the VestGen analysis at an early stage of the disease, because the prevailing paradigm for some decades has been that the vessels simply drop out in early stages and then regrow abnormally at the late stage. So this discovery of the regeneration of more normal vessels at an early stage interests, for example, our new NIH study with an endocrinologist working in the diabetic kidney and retina, because she is globally renowned in adult stem cell therapy. So why she wants to work with us is because hopefully these results will help her develop and fine-tune adult stem cell repair of the retina at early stages before these late blinding stages when there really isn't too much you can do to repair the retina. Well, just to say too that it is these preliminary results that led to our NASA funding, because again with the astronauts, if only about 50% of the astronauts incur these damages after long duration space flight, this kind of analysis may provide more early stage identification of the early vascular changes that must necessarily precede the later damages to the retina, such as visual impairment and cotton wool spots. However, the imaging technology in ophthalmology is rapidly evolving, and really the key companies initiating this reside in our very own Silicon Valley, like Zeiss, Heidelberg, OptiView. So for the first time in the human retina, because of its optical accessibility through the transparency of our vitreous, all the vessels are being visualized. And this is really a revolution in clinical ophthalmology that's happening over the next year as we speak. So working with Richard Rosen and others at the New York Eye and Ear Infirmary Institute, we're beginning to use the VestGen for these kinds of new imaging technologies. And what you can see when we do have the quantification, I'm sorry, I can only really provide the maps in the scope of this talk, but central retinal vein occlusion is a common retinal visual impairing disorder. So we were able to map the vessels and compare them with a normal healthy control, where you see the avascular area of the macula, again, very important for human and primate vision, compared to the loss of vessels in CRVO, and an intermediate state in the fellow eye of this particular patient. And I do want to comment at this point that as I attend these annual ophthalmology research meetings over the last 10 years, I have to say that NASA optical and imaging evolutions for telescopes and astronomy are frequently cited as contributing to the evolutions in this biomedical imaging, including this particular technique, the adaptive optics technique that we studied here. So as I've described, we're starting to embark upon looking at the astronauts pre and post flight, and I can't even predict how that's going to end up a year from now because of bringing in these new imaging technologies that show for the first time all the small vessels. So now, in the remaining period of the talk, I would like to introduce you quickly, just an overview, just pictures are worth 1,000 words of some of the other tissue and organism and disease applications. So we've had about 145 requests worldwide from every continent except Antarctica. Institutions such as Harvard, Mayo Clinic, the US Environmental Protection Agency for toxicology studies, where the biomedical researchers working in head-to-toe vascular-dependent applications such as cancer, diabetes, heart disease, would like to be able to use our software. And this, for me, was a big motivation in coming to Ames because of the life sciences expertise at Ames. Ames is the home of the space biology, leads the space biology for NASA, and also the IT supercomputing expertise here. So Dr. Reinecker, who is an oncologist, immunologist, at Harvard studies GI inflammation and its regulation with really superb intravital imaging. So he sent us his images, and we have analyzed this with the network option of the VestGen, in which you can see clearly the increasing inflammation and really the increasing irregularity and disorder of our normally very regular vascular networks. Similar studies of network tree composites were done with John Sears and a model of infant retinopathy of prematurity at the Coli Institute. And with Michiko Watanabe and others at university hospitals and Case Western, we used the VestGen network, tree network, and mature tree analysis options to chart the development of the coronary vessels from their initial very diffuse amorphous network into transitional structures and ultimately a mature tapering vascular tree around the little mouse heart. Working now here in space biology with Sharmila Bhattacharya and Ravi Kumar Hosamani, we're starting to look at venation in the fruit fly wing, which is a major genetic model. And what's very interesting about this system, of course, it's a major model for the ISS and space biology exploration. But what I find fascinating in this area is that the insect wing is very stereotyped in its vascular patterning. So every fruit fly has essentially the same vascular pattern. This is fundamentally different from vertebrate biology, in which each cam was as unique as a fingerprint. Similarly, for our own human retina, each of your vascular patterns will be as unique as your fingerprint. And yet, the space-filling capacity of these cam and retinal structures are very uniform when measured by a fractal-based analysis, driven undoubtedly by uniform needs within the tissue for oxygen. So now to talk a little bit about blood flow. Mark Burnett is a scientist working in aerodynamics at NASA who developed a particle imaging velocimetry code, his PIVPROC, to chart the flow of particles within the large wind tunnels we see about us at NASA, flowing at Mach speeds of 1, 2, 3. So we used his software in the quail embryo, in this case the yolk sac vessels, not the cam, to look at blood flow intravitally. So I think you can see in this movie the physiological fact that, if I can get up there, the red blood cells, which are the particle markers, are flowing much more slowly in the small vessels before being collected into the much faster flow of the larger collecting vein. So when measured by his PIV software that worked at Mach speeds of 1, 2, and 3 in the wind tunnels, we were able successfully to track the velocity profiles, the overall volumetric flow rates of the blood flow in these little microscopic vessels, and even to the degree of resolving the effect of the pulsatility of the beating heart upon the rate of the blood flow. Of course, it required not only his PIVPROC software, but also his high-speed photronics camera to accomplish this study. So, and then we've used the VestGen now to start looking at Arabidopsis thaliana, a very important space biology and terrestrial model for plants. And again, it's a fascinating fractal rescaling story, as in all of these tissues, because this down here is the actual size of the vascularized plant, which within 6 days has grown to this much more complete and yet ever fractally branching and growing structure. I did train, my final training was at the University of California, San Francisco, in the laboratory of Donald McDonald, who is globally recognized for his abilities to visualize vessels in tumors and inflammatory disease. So many of the techniques I've developed since then in my own research were learned in his laboratory, working on the expression of, in this case, the alpha-5 beta-1 integrin that increases with increasing tumor size. So for those of us working in biology, an interesting phenomenon in the field is that you talk about molecular mechanism, morphological mechanisms, blood flow, even cell biology mechanisms. And these are all are perspectives on a very complex interdependent system that is a living organism. So there's no time to talk about this today, but I just wanted to show to you, as my new space biology colleagues, that we really can, using the imaging technology, modern imaging technology, in which you can visualize the expression of a single molecule, a single gene or protein, via fluorescence microscopy, that this really provides a link between the VestGen vascular analysis and the very complex expression profiles of genomics, proteomics, metabolomics, the other kinds of omics technology that are being now investigated by our new gene lab. So what we are now doing with the plant leaves and in other studies are thinking about incorporating essentially endless number of bioinformatic dimensions of information, along with the Euclidean branching patterns of the vascular morphology. Some of the cell biology mechanisms of the lymphatic system in this case, and the associated smooth muscle that covers the nearby vessels. We did summarize these studies with our model of several cell biology mechanisms participating in various phases in angiogenesis. So I don't think I have time to demonstrate the software today, but we'd be very happy to show it to anyone who's interested. We've got it uploaded on the software. Basically, it presents to the biomedical researcher the three options of a vascular tree network or tree network composite, and then maps and then quantifies, along with your calibration factor, these various types of images. So I think what I've tried to introduce today is the ubiquity of complex fractal-based vascular systems for higher organisms on Earth, including humans. The vascular branching is fractal-based, and I think offers us a system that necessarily integrates very complex molecular and pathway signaling. I think using this research tool, we've made some surprising discoveries. Most often, we merely confirm what you might plausibly hypothesize or what is already known. But then now and then, there are the surprises that indicate important new avenues of research. So just want to say that fractal phenomena in nature is all about us, including the trees that surround us in our beautiful planet. And would like to acknowledge the many people that I've participated with in this research. It certainly has been an honor to work first. This is Patricia Keith and Mary Vickerman, the software development team with whom I developed the VestGen software. This is our new team here at Ames. Ruchi Vyas, Amanda Vu, and Sneha. And also our long-term collaborator continues to work with us on our new studies, Dr. Krishnan Radhakrishnan. There have been a number of biologists, physicians, and engineers inside NASA and at these other institutions who have participated in these collaborative studies. So thank you for your attention.
Thank you very much. Thank you. So we have time for questions. If you have a question, please raise your hand and wait for the microphone.
Hi, Pat. You had mentioned earlier on your presentation something about increased visual impairment in tropical zones. Is that something that you can elaborate on, or?
Well, in the tropics, of course, light is much more intense. So I think it's not so much the retinal problems, but more the opacification of the anterior portion of the eye, the cataracts that develop. But they also are very interested in the posterior portion of the eye, the retina, because of diabetes, age-related macular degeneration, these other vision diseases, which have somewhat greater prevalence in a tropical climate.
So I have a question. So if you look at this software, and you've sort of mentioned it before by starting and looking at basically looking at the atmosphere, have you tried of applying this to other types of fields besides biology, specifically looking at maybe earth science images across looking at planets and other systems like that to see whether this could be applied in that source?
Well, that's a very interesting question. And people have suggested that we look, for example, at arterial branching systems in rivers. When you look at NASA and other atmospheric photographs of the branching patterns of rivers, or if you're just flying over the country in a plane, there's definitely a similarity, I think, with vascular branching. But of course, it's not being driven by our pumping hearts. It is a fluid-based system, however, and that's one pattern that I think has strong analogy. I have to say that I can't think of too many others that have been suggested to us, but certainly you're very right, Dr. Cohen, in the very common presence of not only branching systems, but other kinds of fractal branching patterns in nature. I think they're so common because they just have they have a way of space filling is really the fundamental concept of the fractal field.
Thank you for the talk. The question I have is for low gravity and long flight for the astronaut. I mean, obviously the blood flow, the branches in your eye is the most obvious, but could there also be changes in other parts of the body that we don't know yet?
Yes, that's a very important question. People have been researching the fluid shifts that are known to occur in microgravity for some decades and the impact of that on the human body. This is not, of course, that problem is something that has been known for some decades because in microgravity, the fluid shifts in our body are at the level of 1 to 2 liters and go to the tops of our bodies instead of to the bottoms of our bodies as on Earth. I think it's only with the long duration flights that we have begun to initiate that we're seeing the impact of these adverse impact of these fluid shifts on the visual impairments that appear to be associated with increased intracranial pressure. But again, as we know, mechanistically to prove this and investigate this is very difficult. We may have that hypothesis, but the actual medical studies are very difficult. NASA has made it a high priority. It's a pretty equivalent to the appreciation of bone density losses. So therefore, we are investigating with studies such as ours, but many other studies by ophthalmologists, physiologists in the brain. Also, the hind limb and weighted rodent model is being used to look at these questions of the adverse effects throughout the body of the fluid shifts, as mediated necessarily by the small vessels.
Okay, if there are no further questions, please join me in thanking Dr. Parsons for an excellent seminar.